Hello friends, Ace Botany, next episode, third one, chapter name, Sexual Reproduction in Flowering Plants. And this episode deals with female part, that is Megasporogenesis and Megagametogenesis. Exam point of view, theory as well as NEET exam important, like previous episode. And after watching these episodes, if you are interested, please subscribe or like it, share it, comment on it, so we can proceed. First, I will share the screen. It will be easy for you. I think now the screen came, so we can proceed. Female reproductive part, which is otherwise called pistil or carpel. For the terms will be megasporogenesis, ovule, and embryo sac. So all these we have to discuss today. And each pistil has got three parts. Pistil or carpel, both way we can say ovary, style, and stigma. Ovary is the bottom swollen part. Style is the elongated tube. And stigma is the place where insect will land. So by which pollination can take place. Some aspects of this chapter is closely connected with the morphology of first year. Now when we come to the topic for the detailed manner, we can find in the NCERT book itself 4 color diagrams are given. And there they are explaining 4 basic terms, morphology terms still: monocarpellary, multicarpellary, apocarpus, syncarpus. Single carpel, many carpels, carpels are free, carpels are united. For this, some examples they are given. That is that 3 diagrams of the out of 4 stands. What is a popular question? One, pistil of hibiscus. Pistil of hibiscus, it is multicarpellary and syncarpus. Then pistil of papaver, again it is multicarpellary and syncarpus. Now when we come to Michelia champaca, it is multicarpellary and apocarpus. So first 2 syncarpus, Michelia is apocarpus. After that, 4th diagram, that is dealing with the typical anatropous ovule. That will be observing later. Now when we come to an ovary, ovarian cavities are called locules. And we can also find placenta on these ovarian cavities. Arising from the placenta, there we can observe megasporangia. And ovule is nothing but indegmented megasporangia. Arrangement of ovule on placenta is called placentation. First year we studied. And placenta is made up of most abundant tissue in plant kingdom, that is nothing but parenchyma. So I think first page is clear. All the basic aspects we discussed there. Now we can proceed to further events. Yes. An ovule that is specifically defining an indegmented, indehiscent megasporangia. Many different types of ovules are available. But syllabus-wise, we have to learn only 2. One is most common type, anatropous or inverted. Second is less common and most primitive, orthotropous or straight or erect ovules. So first one, anatropous or inverted. It is commonly seen more than 80% of angiosperms. It is observed. And how it become inverted? Because 180-degree turn for ovule takes place in the sense, so 180-degree turn for ovule, along with it, that makes micropyle lies close to hilum. So 180-degree turn for ovule, then micropyle lies close to hilum. Diagram it is very clear. And second one, that is orthotropous or straight or erect ovule. most primitive. It is commonly seen among gymnosperms, but few angiosperms also possess it, like pepper. And how it becomes straight or orthotropous or erect, we can find micropyle, chalaza, funicle lies in a straight line. In the diagram, you can observe a blue-green line. In both cases, you can observe it. Now, what are the main parts of a typical ovule and how these ovules has been classified? So different types of ovules like hemianatropous, circinotropous, camphilotropous, amphitropous, but that are all not in syllabus. So types of ovule based on the relative positions where micropyle, funicle, and the body of ovule lies. So now parts of the ovule. First is funicle. Funicle is otherwise called stalk of the ovule, and it is attached to the placenta. So funicle, stalk of the ovule, attached to the placenta. Then second one, chalaza. Chalaza is commonly refers to the base of the ovule. Third is micropyle, or a small pore of the ovule, where we can find integuments will be absent. A similar comparison or thing we studied in previous case, germ pores in pollen grain. And what is the function of micropyle? Through this micropyle, pollen tube finally enters. Fourth one is hilum. Hilum is the attaching point between funicle to ovule. So funicle to ovule, that is connected by a point called hilum. Next item is integuments. Integument means protective layer. Two integuments will be there, outer integument and inner integument. Micropyle is the area where integuments are absent. Last one, embryo sac, otherwise called female gametophyte. Now surrounding the female gametophyte, the structure is called nucellus. That is mass of tissue which surrounds female gametophyte, cells will be diploid. So these are the main components of a typical ovule. Almost 7 items. I think idea is clear. Now we can proceed to third one. In this case, you can find megasporogenesis. After that, megagametogenesis. Megasporogenesis mainly deals with the formation of megaspores from megaspore mother cell. First set of diagram like a cellular diagram. Second is more towards a diagrammatic representation. Anyway, first we can roughly analyze what is the procedure. So it starts with or from nucellus. What is nucellus? A mass of tissue which surrounds female gametophyte. Cells of the nucellus will be diploid. From there, a prominent cell arises that is called archisporial cell. 12. And that will act as megaspore mother cell diploid. And that megaspore mother cell will be usually at the micropylar area. Area-wise, it is an area of question. And in megaspore mother cell, we can find meiosis takes place. After meiosis, as usual, 4 megaspores will be formed, and all of them will be haploid. And among these 4 megaspores, only one may be functional, 2 can be functional, all the 4 can be functional. But commonly, only a single one will be functional, and that is towards chala end. Functional megaspore will be on the chala end, that is fourth from micropyle. And in this functional megaspore, one mitosis, followed by next mitosis, followed by next mitosis. So 3 continuous mitosis, or we can say 3 generations of mitosis. Finally, it results in a mature female gametophyte or embryo sac with a 7-celled and 8 nucleate. So that is another famous question. How many celled, how many nucleate in a mature embryo sac? It is 7-celled and 8 nucleate. I think that part is clear. Now we can proceed to next one. Next embryo sac, we have to explain again once more how the megasporogenesis. So one of the cell in nucellus is diploid tissue at the micropylar end or area, will act as megaspore mother cell which undergo meiosis to form 4 haploid megaspores. Usually, normally, common type, only one survives, and that will be towards chala cell end. Undergo mitosis 3 times, producing 8 nuclei. And there will be a rearrangement, and it finally becomes 7-celled and 8-nucleate. It becomes 7-celled and 8-nucleate embryo sac. So 3 types I told you, 1 megaspore only functional, 2 functional, all the 4. So accordingly, there is a classification for megaspore. It is concerned with or connected with the famous Indian plant embryologist P. Maheshwari. And criteria for the classification is number of megaspore nuclei involved in embryo sac formation. We have to learn only monosporic. Typical example is polygonum, bisporic allium, tetrasporic atoxa. So for us, syllabus-wise, only polygonum or monosporic type. Now next is organization of embryo sac. What are the components of embryo sac, or what all structures we can observe there? 1. Synergids. SYN, that itself indicate union. So synergids. Syncarpus, carpels united, again SYN. So here the term is synergids, otherwise called cooperative cell, otherwise called helper cell. So in the case of synergid, we can observe its nucleus will be at the micropylar area, and vacuole will be at the Chala cell area. And it lacks a cell wall at the maturity at the Chala cell side. So nucleus micropylar area, vacuole Chala cell area, lacks a cell wall at maturity at the Chala cell side. And in synergid, connected with it, we can find a structure called filiform apparatus. It is finger-like projection covered by a sheath, and its location is micropylar end, directing the pollen tube towards the destiny. And its chemical nature is cellulosic, or more specifically, it is spectrocellulosic. And filiform apparatus can act as an obturator, giving direction to the pollen tube. And synergids will be usually short-lived. Short-lived in the sense, along with the pollination and fertilization, when its function is over, they degenerate. And what is the function of this synergid? Function is chemotrophic attraction of the pollen tube towards egg or destiny or egg apparatus, what may be. It is going towards egg. So function, chemotrophic attraction of pollen tube towards destiny. Chemotrophic, the term, exam question, chemotrophic attraction of pollen tube. Second structure is egg. Egg, when we start to discuss, its wall thicker at the micropylar ends, and the nucleus at the Chala cell area, vacuole at the micropylar area. So just opposite the previous one. Wall will be thicker at the micropylar end, nucleus at the Chala cell end or Chala cell area, vacuole at the micropylar areas. Egg and synergids have got connection through a structure called plasmodesmata. And this we can compare with the sieve tubes and companion cell in phloem, what we studied in anatomy. Likewise, we can compare pit fields. Here it is plasmodesmata, against synergids will be connected. Third structure is antipodal cells, which is otherwise called vegetative cell of the embryo sac, because in polygonum also there is a vegetative cell. Here, vegetative cell of the embryo sac, female side. And antipodals will be usually 3 in number. It is also short-lived and mainly concerned with the absorbing nutrients from the developing nuclear cells. Surrounding nuclear cells, nutrients will be absorbed. So connected with the nutrition. So antipodals will be 3, synergids will be 2, and there will be a single egg. I think that part is clear. Now we have to proceed to central cell. Central cell, that is one of the largest cell of the embryo sac. And it contains 2 polar nuclei. Initially, that is N plus N. Later, when the pollen tube comes, the 2 N nucleus will get fused to become diploid. And now it is called secondary nucleus or definitive nucleus. That is how the probability changes into diploid, central cell. And central cell is supposed to be the mother cell of the endosperm. At the same time, largest cell in embryo sac also, central cell. So these are the 4 parts mainly we can observe, 4 to 5 parts mainly we can observe in embryo sac, starting with the synergid, cooperative cell, helper cell, egg, antipodal cells, vegetative cell of embryo sac, central cell. So altogether 4 items. Now it is time for questions. Theory question is given. Entrance question also given. 5 each. Later, after finishing all this, I will be supplying both type of question, more in number. So we started with what all we'll be dealing today. Then Megasporogenesis, integumented, indehiscent embryo sac, ovule. Then we proceed to NCERT, 3 to 4 diagrams explaining multicarpillary, apocarpus, syncarpus, all this. Then what is meant by locules. Then placenta, ovules attached to placenta, placentation, parenchyma. This will be discussed in the first page. And the second page, ovule, integumented, indehiscent, megasporogenesis. Then 2 common types of ovule, anatropous as well as orthotropous. Their features, why they are anatropous and orthotropous. Classification of ovules, what are the criterion. Then labeling of ovule. After that, we proceeded to Megasporogenesis and Megagametogenesis, starting with the nucellus and archisporial cell. Then what are the structures seen in an embryo sac, 7-celled 8-nucleate, how it become monosporic, bisporic, tetrasporic, each component, their features. After that, we observe certain theory question and entrance question. So if you like this episode, subscribe all, share, comment, and like it. So next episode, we will continue sexual reproduction in flowering plants, mainly pollination, fertilization, all that area. So we can meet later. Thank you.